This is the second video devoted to enzyme inhibition, to irreversible inhibition. Reversible inhibition is considered in the first video. Irreversible inhibitors bind tightly to enzymes. Their classification differs from book to book. Here we consider four common types of irreversible inhibitors: group-specific reagents, affinity levels, also known as reactive substrate analogs, suicide inhibitors, they are also called mechanism-based inhibitors, and transition state analogs. Group-specific reagents react with specific side chains of amino acids. Diazopropyl phosphofluoridate, or DIPF, reacts with the serine residue in acetylcholine esterase, an enzyme important in the transmission of nerve impulses. DIPF and similar compounds that bind and inactivate acetylcholine esterase are potent nerve gases. Affinity levels, or reactive substrate analogs, are molecules that are structurally similar to the substrate for an enzyme. They covalently bind to active site residues. They are thus more specific for the enzyme's active site than are group-specific reagents. Tacil L-phenylalanine chlormethyl ketone is a substrate analog for chymotrypsin. The compound binds at the active site and then reacts irreversibly with a histidine residue at the site, inhibiting the enzyme. The compound 3-bromacetol phosphate is an affinity level for the enzyme triazophosphate isomerase. It mimics the normal substrate, dihydroacetone phosphate. Suicide inactivators are relatively unreactive until they bind to the active site of a specific enzyme. A suicide inactivator undergoes the first few chemical steps of the normal enzymatic reaction, but instead of being transformed into the normal product, the inactivator is converted to a very reactive compound that combines irreversibly with the enzyme. These compounds are also called mechanism-based inactivators because they hijack the normal enzyme reaction mechanism to inactivate the enzyme. Examples are NN-dimethylpropargylamine and diprenyl, inhibitors of the enzyme monoamine oxidase. Monoamine oxidases destroy neurotransmitters such as dopamine and serotonin. The inhibitors increase the level of neurotransmitters by reducing neurotransmitter decoy. The monoamine oxidase inhibitors are used to treat Parkinson's disease and depression. These diseases are associated with low levels of dopamine and serotonin respectively. Difluoromethylarnitine is a suicide inhibitor of arnitine decarboxylase. The enzyme participates in biological amine synthesis. The normal substrate of the enzyme is arnitine. Difluoromethylarnitine gets into the active site, undergoes some transformations, forms covalent bonds with a group in the active site of the decarboxylase and blocks. Biological amines help to structure DNA. Protist single-cell eukaryotes called trypanosomes cause African sleeping sickness or African trypanosomiasis. They are very sensitive to such types of agents. Difluoromethylarnitine is used to treat the disease. Penicillin interferes with the synthesis of the Staphylococcus aureus cell wall. Bacterial transpeptidase enzyme catalyzes the formation of the cross links that make the peptidoglycan of the cell wall so stable. Penicillin targets the enzyme. It mimics the structure forming in the active site of the transpeptidase made of 2D-alanine residues. Bond penicillin then forms a covalent bond with the serine residue at the active site of the enzyme. This penicillyl enzyme does not react further. Hence, the transpeptidase is irreversibly inhibited, and cell wall synthesis cannot take place. Transition state analogs bind to an enzyme more tightly than does the substrate in the enzyme substrate complex, because they fit into the active site better than the substrate itself. That is, form a greater number of weak interactions. Apart from previous irreversible inhibitors, they need not bind covalently to the enzyme. For example, transition state analogs designed to inhibit the glycolytic enzyme aldolase, bind to that enzyme more than 4 orders of magnitude more tightly than substrates. The human immunodeficiency virus is a causative agent of acquired immune deficiency syndrome. Most of the viral genes are translated into large polyproteins, which are cut by the HIV protease into the individual proteins needed to make the virus. The powerful anti-HIV drugs, called protease inhibitors, were designed in part as tight-binding transition state analogs. The HIV protease cleaves peptide bonds between phenylalanine and proline residues most efficiently. The active site thus has a pocket to bind aromatic groups next to the bond to be cleaved. HIV protease inhibitors share a core structure, a main chain with a hydroxyl group positioned next to a branch containing a benzyl group. You may see this arrangement mimics the transition state forming during catalysis. The benzyl group gets into the hydrophobic pocket of the enzyme when the adjacent hydroxyl group mimics the negatively charged oxygen in the tetrahedral intermediate in the normal reaction, providing a transition state analog. So the drug form multiple weak interactions with the enzyme active site and block it. As for kinetics, if an enzyme molecule is irreversibly inhibited, such as by covalent addition of a specific irreversible inhibitor to the active site, that enzyme molecule no longer can participate in the reaction with its substrate. Thus, the effective concentration of the enzyme in the solution is reduced. Since V maximal is directly proportional to the enzyme concentration, V maximal decrease. The rest of the enzyme molecules that are not affected by the inhibitor will convert the substrate into product, and the same amount of substrate will be required to reach half of the lowered V maximal. So on the Landwehr-Burke plot, it will look like non-competitive inhibition, which is reversible. This perhaps is the most common behavior of irreversible inhibitors. There are some special cases exist, however. How can we distinguish reversible non-competitive inhibitors from irreversible? We can do it, for example, by building up some additional plots like the one shown on the slide. V maximal versus total enzyme concentration. If a substrate is in excess, V maximal depends on the enzyme-substrate complex concentration, which in turn depends on the amount of available enzyme molecules, E total. In the presence of non-competitive inhibitor, we need more enzyme to reach the same value of V maximal. However, even if the enzyme concentration is low, we still can see that the reaction can proceed because some enzyme molecules are free from the non-competitive inhibitor. It is a reversible inhibitor. It can dissociate from its enzyme. Irreversible inhibitors block enzyme molecules completely, and at certain enzyme concentration, the reaction cannot occur at all. If we add more enzyme to the system, all inhibitor molecules will bind to a fraction of enzyme molecules when the rest of the free enzyme molecules perform a normal reaction. So you can see that the slope is the same, but the intercept with x-axis for the irreversible inhibitor is shifted towards higher enzyme concentrations. Thank you for your attention. See the info concerning reversible inhibition in my previous video.